Hi guys! Well, the question today is how is neutralization involved in host defense? It is needless to say, when referring to host defense, we're talking about the protective features of antibodies. Immunity mediated by antibodies is known as humoral immunity. And there are three main ways the antibodies deal with the various pathogens and their toxic products: neutralization, opsonization, and complement activation. But today, our sole focus will be on the first, neutralization. Neutralization, as the name suggests, is the mechanism by which toxic products from different pathogens are neutralized by the work of antibodies. The simplest and most direct way in which antibodies can provide protection from pathogens or their toxic products is by binding to them and thereby blocking their access to the cells that they might infect or destroy. Normally, the unbound toxins entering the body can easily react with receptors on the host cell, whereas the toxin bound to the antibodies, forming a toxin-antibody complex, simply cannot. Antibodies neutralize complete virus particles and bacterial cells by binding to them and inactivating them, and in this way preventing entry into cells and also preventing replication. The new antigen-antibody complex is eventually scavenged and degraded by macrophages. Macrophages are specialized cells involved in the detection, phagocytosis, and destruction of bacteria and other harmful organisms. So, just to go over this in a nutshell, we have our antibodies ready to take action as a pathogen or a toxin enters the body. These are efficiently neutralized by the antibodies, which create an antigen-antibody complex that can no longer enter the cells. Macrophages then come into play and engulf the antigen or toxin and successfully provide protection to the body. So there we have it, a clear picture on neutralization in host defense.